As always, I appreciate you for coming today. This is the March 30th meeting of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. It seems that week by week, we get further and further from the title of the working group, but they're always exciting and interesting talks. I need to remind you that, as always, this meeting is live streamed and recorded and available on YouTube, so please be aware of that. Tom Helikar and I are always glad to take your suggestions, comments, and criticisms. Jim Sluka handles the back end of things and also ideas. Lorenzo handles the schedule, and Joel has been helping out over the years with a variety of things. So we're all available for suggestions and comments. Please help us raise our profile and distribute what we do, especially our YouTube channel, but also the IMAG Wiki page. And we have a LinkedIn page and a Twitter feed. The Twitter feed uses MSM Viral as a tag. Please help us get the word out. There are some announcements. I got in the mail the other day a review copy of a book related, I believe, to the VPH. I think Peter is the head of high tech and performance computing for VPH, with a wonderful title. I'm so jealous that they took that title, called Virtual You, How Building Your Digital Twin Will Revolutionize Medicine and Change Your Life. If you actually read the book, I think the perspective is rather different from the one that we take. And so it's quite interesting to see what they've done in there. Because it's quite a different perspective. I think it's a little bit more what I would call virtual tissue-y than digital twinny. But it's quite interesting. It's Princeton University Press. The ISBN number is there. And I thought, since they sent me a copy, I would let people know that they had published it, and my people might be interested in seeing that perspective. As always, I want to remind people about the imminent big workshop in Paris. I think a lot of people are going to be there. I know a large number of people I see here I know are going to be in Paris, at least for a week or two. I'm going to be there for the first 2 weeks of the meeting. In May, it's getting closer and closer. I believe that they are not completely full. So I think if people want to go, I think there's still opportunities for you, students, postdocs, and so on. I don't, Anna and Reinhard aren't here today to comment on where they are or the organization. We've talked about possibly trying to arrange a discussion panel for this meeting, May 25th, out of Paris. We'll see whether that works. That would be fun if it happened. The plans for the IMAG anniversary meeting in Bethesda are coming along. The organizers are Shane Pierce-Kotler, who's spoken to us a couple of times, and also Herbert Sauro. So if you're interested in learning more about that, talk to them. Talk to, of course, Grace Peng and the others at NIH who are setting this up. I think it's quite potentially quite an interesting meeting. I want to remind people that we are still doing an open search for faculty in this general area. And I want to say that the digital twins could be understood very loosely. We've had a little bit of additional direction from our central administration that they would prefer to hire at the senior professor level. So if you're a full professor and you want to come to Indiana, or you're an associate professor who wants a promotion to a full professorship, and you want to come to Indiana for that, we have 2 faculty lines available in our Intelligent Systems Engineering department. And we would love to have you join our effort in multi-scale modeling and prediction here. So if you're interested yourself, I'm sure anyone in the room here would be great for this. Or if you know people who might be appropriate, please let me know. I'm happy to talk to people in confidence about the parameters of this search. I'd be delighted to have you. I have the reminder that I believe that the UBC search is also ongoing now and might be relevant to people here in the room. After a great struggle, Tom Hellikar does have the Glimprint website in preliminary version up. Thanks to everybody who contributed to that. That's always a work in progress, so people please take a look at it. Maybe we can put the link in the chat, although I think if you just search Glimprint, you'll pull it up. Please help us populate that website, correct mistakes, make suggestions, and so on. I realize that it's a big job to create a new website, but we would very much appreciate your input and suggestions for that. We're not going to have a steering committee meeting tomorrow. We felt that people had enough meetings at the moment. So the steering meeting tomorrow is canceled. We will meet in 2 weeks. Are there any other announcements before we get to the seminar? No, in that case, I'll just come back to the schedule. Next time, on April 13th, we have Mikhail Benson from Karolinska talking about the very large Swedish Digital Twins Initiative. April 27th, we have Claudio Montero coming back to talk more about the wonderful Ginkgo modeling platform for in silico simulations. David Basanta from Moffitt will be talking about his human digital twin models for cancer therapy development and optimization, which should be exciting. Megan Haas from University of Virginia, June 8th, will be talking about her work modeling estrogen effects on muscle recovery after injury, which is also very interesting. It's not quite as personalized in the sense it's not individualized to a single patient, but it is studying age variation and hormonal variations effects in great detail. We have still a slot on June 22nd if somebody wants to suggest speakers. And as I say, the May 25th slot may be a live stream or a discussion stream from Paris, which is why we're holding that at the moment. Any other comments or suggestions? As always, we would appreciate your ideas about speakers. Please do come to us and let us know what you would like us to do. Okay, I won't take any more time. We're very pleased today to have Daniel Dimitrov from Heidelberg talking to us about knowledge-based machine learning to extract molecular mechanisms from single-celled and spatial multi-omics. And I will turn the screen share over to you.
Yeah, thank you. I assume you can see my slides now, right? Right, okay. Yeah, well, thank you for having me, first of all. It's a pleasure to be here and to present. And yeah, I think I will do my best to fill in the shoes for Julio, but as you can note, yeah, you can notice it's probably not him, so I'm a bit, yeah, just a PhD student. I'm a third-year PhD student, and my focus of study has been cell-cell communication. So even though I will try to represent most of the tools that we have and the project that we have ongoing in the lab, there will be a bit more of a focus on the things that I'm doing currently, and I hope that you'll find it interesting. So yeah, so just to begin as a general introduction of the lab. So we are a purely computational lab. We're here in the University of Heidelberg, and as many other computational labs, what we focus on usually is trying to disentangle insights from data and to obtain certain associations or predictions when using omics data, right? And I guess what makes us a bit So what is specific to us is that we also rely a lot on prior knowledge, and there has been a lot of effort going on for long before I have joined, basically trying to standardize prior knowledge so as to expand prior knowledge resources. And the whole idea of this and the whole lab culture around this is that we believe that the use of prior knowledge can first reduce the dimensionality of the data, and hence increase statistical power, while at the same time also kind of allowing a bit more of a biological or mechanistical interpretation of the large-scale data that we usually work with. And so you will notice that also a lot of my talk will be focused around prior knowledge, or at least indirectly focused on using prior knowledge in trying to extract social communication insights. So I think, as I mentioned, there has been a lot of effort trying to standardize prior knowledge, and maybe the flagship of the lab when it comes to this is this project called Omnipath. And what Omnipath is this, let's say, rich database of mostly focused on protein-protein interactions that has over 100 knowledge resources for basically ranging from pathways to transcription factors, and now we're also expanding it in the direction of metabolites, as well as considering certain directions when it comes to microbiome. But these are still ongoing developments. And another thing that is currently happening with Omnipath is that it's being, I guess, a bit more technical, but it's being transferred onto this new way of, I guess, data biological knowledge representation, which has BioCipher, a project that was recently, a manuscript was recently posted to archive, in which we show, or Sebastian Loventanzer actually shows how proposes a framework with which knowledge representation can be better standardized. So in the end, this is a database which can be used to standardize biological knowledge, in the sense that it was applied to federated learning, for example, or it has basically the whole idea is that it's highly modular, and this you will see is also a recurring topic when we speak about the tools of the lab. So maybe just to give a simple example of another tool, and I guess again this type of modularity that we try to achieve is, and perhaps one of the simpler ones, is this tool called decoupler. And what decoupler does is that it's just a ensemble of enrichment analysis methods, and it also is integrated with this Omnipath database, and hence has let's say direct access to multiple pathways as well as methods to do enrichment analysis. So this is just as an idea of the things that we're doing. So to go back to Omnipath, Omnipath is of course not just limited to, as I mentioned, to intercellular, so things that happen within the cells, but it's actually also focused on intercellular knowledge. And this is how, I guess, the project that I will mostly focus on happens. So when we think about intercellular knowledge, so interactions between cell types, usually what comes to mind are canonical, like the most canonical thing is the secreted signaling, right? So ligand receptor interactions. And this is this idea how it started, at least for me, is that, so we have ligand receptor interactions, and we assume that these are, let's say, a decent representation of cell-cell communication. And recently, there has been, at least when it comes to single-cell RNA-Seq transcriptomics, when it comes to cell-cell communication, has become like a hot topic, and it's not surprising given that it's important in homeostasis, development, and disease. Of course, the immune system has a profound role in this, and largely this has been driven by the technological advances, and now lately a key term to which I will come back to has been termed, which is these multicellular programs, or in other words, coordinated events which occur between cell types within a tissue or a system. So as I mentioned, when we think about communication inference or cell-cell communication inference in single-cell data, we usually imagine this very simplistic approach, right? So we do one-to-one, so basically one-to-one predictions between a source, so a cell type that is expressing a certain ligand, and a target, which is expressing a certain receptor. And we assume that this expression actually represents communication. Whether this is truly the case, I believe we will come back to. But I think just to make sure that this is more or less, yeah, we have wrapped our heads around this, is that we somehow assume that communication first occurs at the cell type level, and second, that it's somehow represented by one-on-one relationships between cell types, right? So is this communication? I'm not sure, right? So we will come back to this. So, well, back when I was starting my PhD, how this usually worked was that there were a lot of new tools to infer cell-cell communication that were coming out, but usually they came in a fixed state in which you had an interaction prior knowledge resource, and then you have a method that was also attached to this resource. And I guess our simple assumption here was that perhaps we should see how much of an impact does the choice of resource and method make. And that's why we developed this tool called LIANA. And it's really simple, right? So it grants access to different resources via Omnipath, and it also provides a bunch of methods as well as their consensus. So using LIANA, what we did, and I guess this was published last year, what we saw was that basically when you run different methods with different resources, I realize this is maybe a bit of a complicated plot, but all you need to see is that here the Descartes index, when we look at the highest ranked interactions, was rather low. Or in other words, the agreement between tools was quite low, right? When we look at different resources and methods on the same data sets. So I guess this is not something that is unique to this field. This is a common thing that occurs in computational biology, that different tools don't usually agree. But nevertheless, we still found it a bit concerning, and we asked the logical question, right? So do these tools, despite their disagreement, actually capture some biological signal, and on top of all their assumptions? So here, as I mentioned, there are a lot of assumptions when it comes to these tools, and specifically, we assume that somehow all events that precede the interaction between two proteins or two cell types are somehow also occurring, right? And we do this by expression alone. So we assume that translation has happened, the secretion has happened, and so did the interaction between the proteins, and then there was some kind of an intercellular response. So logically, we wanted to evaluate them a bit, those methods, and see if there was indeed some signal being captured. And here, a major limitation being that there's no ground truth, which again is a common occurrence. So we had to, let's say, generate our own inferred truth. And in this case, we focused on spatial colocalization between cell types, as well as downstream cytokine activities. I will not go into detail into this, but this type of, these two benchmarks are now being, let's say, integrated into the Open Problems initiative, which is let's say, a living benchmark that keeps evolving. And the idea is that everyone would be able to then benchmark their tool in a relatively simple manner, in a way that is standardized to every other tool. So it's basically shaped by the developers of the tools, and it's aimed to be a fair platform. I can come back to this if there is interest in the specifics of the results, but the whole conclusions of these benchmarks that we are doing currently in the Open Problems as part of open problems, but also the ones that were previously published, was that more or less we do capture signal, some biological signal, but really we cannot say which is the best method, and we really cannot say, let's say, is this interaction actually happening, or is it co-expression alone? So this is something to keep in mind. So just what they presented so far is perhaps like a bit more simple side of LIANA. So the tool that they developed, and it's just this tool that combines different methods to infer cell-cell communication events in single-cell data and resources, right? So, and as I mentioned, it's being integrated into this platform in which it should be, let's say, provides like standards on which we can improve. So logically, as I mentioned a bit earlier, these multicellular processes or coordinated processes that occur are not going to be studied by the tools that, for example, are implemented in LIANA alone, right? So if we just focus on the interceptors that are being inferred in steady-state data, not sure how interesting this is, right? So I mean, as a tool developer, I'm not certain what are the exact biological questions that this might answer. So to me, where this becomes a bit more interesting is when we think about biological contexts, such as disease or time series. And here we've been working on a, let's say, joint manuscript, which is a bit more focused to be a protocol type of paper with people from Lewis Lab from University of California. This is Eric and Raj, and their tool, which was recently published, Tensor cell-to-cell. So I will go into a bit more detail in this, but before that, I just want to go back to this idea of having this going away from pairwise associations between cell types, but rather trying to think a bit more in a way of a tissue-centric understanding of disease, or disease and communication in general. So there is now a lot of work that is going out that's, well, not a lot, but a few methods that were going out that try to infer coordinated events across cell types in single cell data in an unsupervised manner. And for example, recently there was a paper from Ricardo Ramirez from our lab, which basically repurposed the framework called MOFA. I believe most of you most likely know it, to basically extract this type of multicellular processes. And he then also mapped them into space. So basically inferring signatures, so coordinated signatures from single cell, and then trying to map those into space. I think we can also come back to this if there is interest. But just for the sake of time, I will continue with this idea of combining cell-cell communication inference with factorization approaches. And this is what essentially LIANA and Tensor cell-to-cell do. So it's just a network which allows the user to make a choice of the method that they want to use, resource that they want to use, and it's now described in depth, and it should, in theory at least, work with any design complexity, and it allows the decomposition of intercellular insights per sample to be basically, decomposed into factors that are associated with changes between samples, for example. So just to show why this is important, when you think about running any inference methods, single cell communication inference methods, by sample you get this type of plot, right? And this is how it would look, and this is only 3 interactions. So we're already talking about something that is very difficult to actually make sense of, right? I think this, at least I cannot. So this is why we more or less teamed up and collaborated on this project to bring LIANA and Tensor cell-to-cell together with the idea to perform this tensor component analysis, which is similar to NMF in this case. And what it does in reality is that we represent the sample, the interactions that occur by sample into a higher order tensor, and then we we decompose those into a certain number of factors, and we can then find factors that are associated with a given condition, right? And these conditions, yeah, can be rather flexible. And I think the good thing here is that we can also, the way that tensor cell-to-cell works is that we get, for each factor, we get 4 vectors, and each vector represents the loadings or importances for the samples, for the interaction that occur, that are important for a certain factor, the sender cell types and the receiver cell types. So just as a simple example, I think maybe one of the simplest is just, I will just show PBMCs coming from 8 lupus donors. I think I'm happy here to share the reference because it should have been lost. But it's just 8 lupus donors before and after interferon beta stimulation. And this is how the UMAP looks, right? And when we decompose those, we basically get a number of factors. This works, the number of factors works similarly to how you would do k-means, right? The estimation, get an elbow curve, try to find an optimal number of factors. And then, for example, in this case, we find that factor 5 seems to be very different between the contexts of samples. And you can see, and we can now focus on the ligand receptor loadings or the ligand interaction loadings that are kind of driving the differences between samples in this case. And already, even in this case, we still think we're still working with thousands or maybe 1,000, maybe a couple of thousands of interactions. So we can take this a step further and reduce it into, let's say, into the communication events that are associated with a certain pathway. And in this case, I use a pathway database from the lab called Progeny, together with the enrichment analysis tool called decoupler. Right? So the same idea can also be applied with any other type of factorization, such as MOFA, and it can also be supported with, I guess, a bit more standard differential expression analysis. And I think, as you have seen, maybe, is that, you know, when we work with single cell data, we kind of lose this. Well, we lose the organization of the tissue, right? As soon as we dissociate those cells we no longer actually know their location in the tissue, and we cannot infer that what is like the events that are occurring in a certain proximity, right? So I guess a natural extension here of the LIANA tool that I developed was to also implement those certain number of methods. And in this case, I was heavily inspired by, let's say, SC-hot and spatial DM. I think I should mention this. And yeah, just to represent this, if we have two, so the way that these methods usually work is that they work with bivariate features. So the bivariate metrics, just for example, Spearman correlation or Pearson correlation. In this case, I'm showing local Morans R and its p-values. And what we do with this is that we use a certain proximity weight. In this case, it's a radio kernel with gene expression of 2 genes, and we can see the locations in which certain interactions occur. By the way, there is a lot of things going on in the chat. I'm not sure if I'm not going to answer. I guess it's just links. So thanks for sharing. So here we can see, for example, that this interaction seems to be associated with the fiber tract. And of course, this is Visium brain, so it's quite nice and structured data, so we can easily go back to this. I think it becomes a bit more challenging when we think about, let's say, more chaotic tissues such as cancer, but we can think about this. We can discuss this also. And here, again, as I mentioned, the idea is that, again, we take the existing metrics and we make a framework that is flexible and allows the user to make a choice of the metrics that they want to use. So here, I guess, I show a simple example of ligand receptors, right? But as I mentioned, I'm not sure if communication is driven by ligand receptors alone, right? To me, communication is a complex mechanism, and that's why the whole idea of this framework is again so that X and Y can be replaced by any other cell type. There are certain projects going on in the lab, at least pilots, with immune repertoires, with metabolites. It can be also replaced with pathways and microbiota, which I will show us a brief example of. So as I mentioned, these cell-cell communication events are, to me at least, are a bit more something that is driven by a large number of factors. So it's the patterns of the tissue or the cells, the tissue structures, as well as, let's say, the frequencies that occur in the tissue. So pathways or any other signaling process that can be there. So it's not just two proteins interacting, right? It's a lot of pleiotropy, it's a lot of crosstalk. So we should really try to strive for modeling in this way. Developments that are related to other developments in the lab, which include the Spatial framework, which is an ETL to extract knowledge, to extract knowledge and build knowledge graphs in a standardized manner. There is Corneto, which is a framework which is a bit more networks focused, that is trying to optimize certain algorithms that are already network algorithms that are already in the lab. And there is also other directions of this, which include enrichment analysis of or connecting these with a bit more mystic insights, as well as trying to infer metabolites, for example, from expression data. And that's more or less it. That's my talk. I hope was clear, that we didn't rush too much. But the whole, I think I want to again emphasize the idea of LIANA, and I think that it really represents the culture of the lab, because it's focused to be user-friendly, it's focused to be flexible, and we're doing our best to make it efficient. And we really do try to make everything modular so that it's so basically it can be used by a person with less experience, but it can also be adapted by anyone with a bit more experience into a bit more complicated pipelines. And with this, I would like to thank everyone that has been involved and has been working with me. Of course, I would like to thank everyone in the lab, and I would like to thank you for your time. And yeah, I'm now open to questions. Yeah, thanks.
Maybe leave up the slide which showed the different tools. So that would be good. That one, the one before that, where you had the map of the tools. That might be a good way to frame that one. Might be a good way to frame the discussion or the start of discussion.
Yeah.
Questions, please.
I have a quick question. In your data resources, you had the connectome, and you got various other things. Is any of those databases related to drugs and their effects, drugs and their receptors, drugs and their diseases they're used for?
So I think yes, but most likely it's a bit more. So not so much to this project, but for example, in BioCipher, there is an adapter or let's put it like an independent repository that enables you to, for example, query open targets, the open targets database. So this might be one link to it. And I believe there is also guide to pharmacology, which is also in Omnipath and should have some interactions of drugs to proteins or receptors at least. Well, Carnival, actually think, I'm not sure how many people are familiar, and this is not my area of expertise, but it's a integer linear programming algorithm that tries to find a sign coherent short, well, in reality, a sign coherent smallest sign coherent network. And this has been applied to multi-omics. So for example, there is this Cosmos approach, which tries to, so basically extends Carnival to transcriptomics, phosphoproteomics, and metabolomics. So a joint essay. And this Cornetto network that is I'm currently showing at least the logo of is trying to optimize this so that we can optimize this further so that it's usable without simplex algorithm, and it's also in Python, so it's would be a bit faster in the end, I suppose. And it's just trying to really do this type of modular way of representing also network analysis.
Great, thank you. I also have a second question. I think did not completely understand. Were you using MOFA Plus as a comparative tool, or how exactly? Can you just explain more about how exactly you were trying to talk about?
So I think I should have a slide somewhere here maybe that represents it a bit better. But so I think this might be a bit more confusing than actually helping. But in the end, what Rico did, so Ricardo, what he did in his work, which maybe is a bit more intuitive, is that he represents. So we think about a single cell atlas in which we have multiple cell types, we have multiple patients, and what he does is that he represents each cell type as a view, so rather than as a omics layer, and he can then learn those joint programs across cell types that separate the patients. So this is more or less how it was used, and this is also how I use it here to kind of separate patients, for example, when we think about event receptors in this case. But yeah.
Thank you.
Yeah, can you elaborate on this causal path and Cornetto? Is Cornetto a graph theory application?
I'm not sure. I think I'm probably not the best one to ask for this, since I'm a bit more on the side that is less focused on networks analysis. I'm not sure if I can give the best answer, but the whole idea behind Cornetto is really to provide like a modular framework that we can kind of call the network bus, right? So you can do, I think the idea is that we can be able to do, for example, something like a bit more diffusion focused, or then like walks with restarts, as well as a bit more on the linear programming kind of things. But I don't think that I am really like an expert in this to give you a better answer than that. So I hope it's sufficient.
But the neighborhood to causal path, you're going to interpret pathways in order to reveal some causal relationship via this, what you call modular or diffusion, but it looks to me from the picture, it's like some graph theory.
Yeah.
Okay.
Yeah.
I'm wondering if you have publicly available videos that demonstrate how to use LIANA.
So I don't have videos, but I think I have done really, I've put a lot of effort in providing well, user-friendly tutorials in both Python and R. And as I mentioned, now we have also done an in-depth protocols type application of LIANA and this Tensor cell-to-cell approach. And maybe I can even copy a link right now. I think it's, but it's, yeah, videos are certainly something that we should consider in the future. But in the meantime, I will post, we'll post this link to this. It's still a work of progress. This thing is still unpublished. We hope that it should be, it's already ready to use, but maybe we need a bit of time. But it's all the details are basically there, and I hope it's helpful.
Okay, thanks.
Yeah, no worries. And also LIANA Python, in case Python is your preference.
Okay, other questions? Tom, how did?
Yeah.
Go ahead, Lorenzo, go ahead.
I have one more. I see now in the panel that in the spatial area, well, I picked the name that I'm most familiar for, the Moransar. My question is about what is the general approach of that without too many details? I appreciate that. Is some, I don't know, you pick a gene and then you do the Morans for that particular gene, or you have some other approach where, I don't know, you build a tensor of some space and then you use the intensity of the tensor for that. So is univariate, multivariate, is pseudo multivariate.
It's bivariate, so it's two, so it's X and Y. But also, I think yes and yes, also on the previous two questions. So basically, the way that I see at least the application of LIANA and how it would be written as a manuscript is that, so to me, combining the two technologies, so single cell and spatial, is probably the way to go, since at least when we think about Visium and this standard Visium and dissociated data, since you can use, let's say, the resolution that you have from single cell to infer the, let's say, important ligand receptor interactions that are driving separation between samples or basically driving disease. And you can then use those approaches to map these interactions into space. But so the approaches such as Moransar. But to me, it's also perfectly, so another direction that one might take is that you could also infer all ligand receptor interactions or any kind of other communication representation that you have as an idea. So metabolite, receptor, etc., whatever you have in your in your spatial data. And then one can again apply those factorization approaches or anything like simpler, right? So yeah, like any standard differential expression analysis to really look for differences within niches, for example. So niches here, when I say niches here, I refer to, let's say, clusters of either communication or certain cell types within space. Right. Yeah.
Thank you.
Yep.
Other questions? I guess I'm thinking of Tom, of course, who is the pathway modeler. And I'm always interested. I mean, it's not the job of pathway inference to be telling us what the parameters are. But I'm always thinking about how do you connect the inferred pathways to an instantiable model that you can actually execute. And I wonder whether some of the correlations, the spatial correlations, might not only tell you about the presence of connections, but be able to give you weights. Maybe you already said that and I missed it because I'm not a specialist, but can you get inference of potentially the strength of connections as well as the presence or absence of connections?
Yeah, I think it's a good point. Actually, I mean, this is probably something that will be beyond my PhD, to be honest, when it happens in space at least. But LIANA is already setting the let's say the path of how we actually build those causal paths in space, right? Pun intended. But so the idea here would be, at least, like, let's say a simple scenario would be that you infer, like one would infer the ligand receptor interactions that are important, and then one can similarly just, let's say, like my limited understanding, of course, in modeling causal paths here would be finding, let's say, the shortest causal paths between a ligand receptor interaction and a downstream TFs, or a ligand receptor interactions and multiple downstream TFs, which, from my understanding, again, is probably not as trivial as it sounds. So yeah, I hope. Yeah, and again, like here we take spatial information into account. So spatial distances are weights that we use for the inference of for the relationship between two genes, to also answer the question.
So one question then, I mean, if I have a consistent data set, and I'm sure you said this and I missed it because again, this went pretty fast for me.
Yeah.
I need to listen to this more times and read your tutorial, which looks very good. Is if I have a consistent data set like spatial RNA-Seq, I think I understand how you use that. If I have multiple data sets, suppose that I had different, suppose that I have my omics data, but I also have a map of some other attribute of the cells that's spatially correlated. How do I combine multimodal data to do this kind of analysis? Because there may well be things that, about, for example, cell behavior, stage of cell cycle, the cell is necrotic, something else which doesn't show up in the RNA-Seq, but that might be important to correlate with the things that you're looking at here. Is there a way to layer your channels so that you can have multiple types of data that are integrated in your analysis?
Yeah, I think so. I really like the question. I think, yeah, usually so far I think now it's more or less like the time, at least from my knowledge, in which we actually start to get multi-model spatial data. So this is still something that is to be answered. But how I would imagine it, and this is like something that just comes to mind, and maybe I'm overusing MOFA here, but the same way that one can use a view or a niche, let's say a niche in which we have gene expression, we can similarly have the, let's say, protein abundance for the same niche modeled as a separate view, and these can be basically added indefinitely, right? This is the, I guess, the power of those multi-view type of factorizations.
I mean, I'm thinking, for example, in toxicology, where your networks and pathways, some of the components are molecular species, but other components could be cell behaviors, cell states, changes in particular phenotypes. And so you don't only have species connections. And so to me, it's always hard to understand how to bridge between a molecular concentration and a behavior.
Yeah.
But I think in principle, if it's labelable, these methods should be able to do that.
Yeah.
Can I ask one technical question, or the time is up?
Please. No, Julie, go ahead.
So when you are, you nicely showed the spatial graph and the sort of rule between the genes. I think I missed, when you're analyzing the spatial data, are you doing the clustering of the single cell data separately, or you are directly only looking at the spatial map and the interactions between the genes?
Yeah, I think in this case, I represented only like an independent sample, just as an example. But indeed, if I was to actually do that, and I have, let's say, I have a cohort, like idealistic scenario in which we have single cell data for the same patients, and we also have spatial data from the same tissue again, or neighboring tissue, maybe even easier if we're working with mice, then indeed what I would do is probably focus first on the events that occur in the single cell data and try to map those into space. Cell type, deconvolution, etc.
Yeah, I mean, eventually I hope we get to the extent where in the spatial data, you only get the single cell level resolution. But I see your point.
Yeah, I think I can actually, I think this is a point where I share a paper by Rico. So the one that I presented briefly, but also I think it might be of interest exactly on that topic. So this is the application that he did with MOFA, and also the data that he uses is actually data that was recently published again with the lab in collaboration with Mahen. And in there they have this idealistic scenario of joint samples from the same patient, single cell, RNA-Seq, and space. So I think it's really an interesting application.
Thank you.
Any other final questions? We've had a great discussion. Okay, well, I want to thank you very much for the presentation. We really appreciate it. And we look forward to seeing how this develops. Maybe have you come back, maybe have you give us a tutorial on how to use it.
Yeah, happy to. Yeah. Thanks for having me.
Really appreciate it. Thank you, everybody. Remind you that there is no steering committee tomorrow. We'll have steering committee in 2 weeks, and we'll see you in 2 weeks. Thank you, everyone.